We have learned the structure of plasma membrane and also the characteristic of plasma membrane in the previous chapter. Now, let's look at how substances they are able to cross the plasma membrane in and out. First of all, we know that the main component of plasma membrane or cell surface membrane, they are formed by protein and phospholipid. Phospholipid molecules are arranged in bilayer, in which the hydrophobic tail or the hydrocarbon tail positioned at the middle of the plasma membrane. And the plasma membrane, the thicker portion of plasma membrane is formed by the hydrophobic tail. Hydrophobic tail is not soluble in water. By this, those water-soluble particles or charged particles, they are unable to cross the plasma membrane directly. Only non-charged particles or very small particles like water molecules, they are able to directly cross the plasma membrane. At the same time, those molecules which have very big size, or we call as macromolecules, such as glucose, amino acid, they are also unable to cross plasma membrane directly. By this, what are the methods can be used by the particle to cross plasma membrane? There are 4 methods. The first is the passive transport. Under passive transport, you have diffusion, or we call it as simple diffusion, facilitated diffusion, and osmosis. The second method is called active transport. The third is endocytosis, and the fourth is exocytosis. Now, first of all, let's look at what are the differences between the passive transport and also the active transport. In passive transport, is defined as the transport of substances from a region of higher concentration to a region of lower concentration. Imagine, there's someone who is doing cycling down the hill. At this part is the region of higher concentration, which means at this region it represents the part where you have a lot of solid particles. While this point is the region of low concentration. Imagine it's just like the part where you have very little solid particle. So when the transport substance from the region of higher concentration to the region of lower concentration is just like cycling down the hill. When you are having cycling down the hill, it do not need much energy. By this, in passive transport, when the substances are moving from the region of high concentration to the region of low concentration, energy is not required in this process. By this, it is known as passive because no energy is required. Under passive transport, you have 3 types. Now, let's look at the first one. Diffusion or simple diffusion. Diffusion is defined as the net movement of particles from the region where they are in higher concentration to the region where they are in lower concentration until equilibrium is achieved, which means both components, both regions have equal amount of solid particle. What you need to know is very simple. First, you need to know the definition for each type of transport. Second, you need to know what are the example of substances across the plasma membrane use different type of transport method. So, in human body, transport of gaseous exchange, oxygen and carbon dioxide that happen in the alveolus is an example of simple diffusion. This picture showing the structure of alveolus, and this part showing the structure of capillary. For the oxygen to move from alveolus into blood capillary is a movement of simple diffusion, which means the oxygen can just simply directly passing through the phospholipid bilayer of the cell and enter into blood capillary. At the same time, carbon dioxide can also just directly passing through the phospholipid bilayer of the cell, moving from the capillary into the alveolus to be expelled from the respiratory system. So the gaseous exchange that happen in the alveolus is the example of simple diffusion. What are the factors that may affect the rate of diffusion? The rate of diffusion increases with heat, because when you providing the system with heat, it increase the kinetic energy of substance. So higher kinetic energy allow the particle to move through the phospholipid bilayer faster. The rate of diffusion increases in large surface area. When you have more surface area, more region for the particle to go in and out of the cell. The rate of diffusion is proportional to the concentration gradient. Concentration gradient is just like the gradient of the hill. So when you have higher concentration gradient, the rate will become faster. The rate of diffusion is inversely proportional to the size of diffusing molecule. When the particle have larger size, then the particle have to slowly move through, squeeze through the gap between the phospholipid. By this, the rate of diffusion will be slower. The rate of diffusion is inversely proportional to the distance over which it takes place, which means when the distance of the diffusion is longer, the rate slower. In example, which means when the plasma membrane is thicker, the thicker plasma membrane will need it will take longer time for the particle to cross. The rate of diffusion is also affected by the permeability of membrane to diffusing substances. Some of the membrane, they are selective permeable. Only certain particles can cross the membrane directly, while some cannot. The second type of passive transport, which do not require energy, is osmosis. Osmosis is defined as the movement of water molecule from an area of higher concentration of water to an area of lower concentration of water across a semi-permeable membrane. I want you to pay attention on the definition of osmosis. Look carefully. It's talking about the area of higher concentration of water. The definition is talking about concentration of water, not concentration of solute particle. So in osmosis, it means that when the region have more water molecule tend to move from the region where they are large in number to the region where you have very little water molecule. As you can see in this picture, the chamber in your left hand side, the one in blue color, is the water molecule. So the one in the left, they have more water molecule compared to the chamber in the right. The chamber in the right, they have just very little blue color molecule. The blue color molecule is the water molecule. So by this, water molecule will move from this region where they are in higher concentration of water to the region where they are in lower concentration of water. However, when you talk about solute particle, it is always in the other way. When the region have high concentration of water, always they have low concentration of solute particle. The one in yellow color is the solute particle. So you can see the chamber in your left, they have a lot of water molecule. By this, they have very little solute particle. But the chamber in the right, they have very little water molecule. By this, the solute particle, they are in a lot. Osmosis have three terms: isotonic, hypotonic, and hypertonic. Tonic mean concentration. Iso mean equal or same. So imagine you have a chamber A and B. Chamber A and B, they are connected through a semi-permeable membrane. The semi-permeable membrane only allows passage of very small molecule like water, but it is not allowed the passage of large particle, example like sucrose. So when the both chambers side by side, they have equal concentration of solute particle. For example, the one in your left, they have 2 molar of sucrose. The one in your right, they have also 2 molar of sucrose. So we say they are having equal concentration of solute particle. This is known as isotonic. Hypotonic. Hypo mean very little. Tonic mean concentration. Hypotonic, very low concentration of solute. Hypertonic. Hyper mean too much, a lot. So in hypertonic, it means the solution have a lot of solute particle. So in hypertonic, it means the solution have higher solute concentration. So isotonic, hypotonic, and hypertonic is always in comparison between two solutions. For example, when I have red blood cell, when I place red blood cell in distilled water is a hypotonic solution. By this, water molecule will diffuse from the outside into the red blood cell, make the red blood cell to burst. When red blood cell bursts, it releases hemoglobin. By this, the bursting of red blood cell is also known as hemolysis. However, when I take the red blood cell, place them in a concentrated salt solution. This will cause the concentrated salt solution is hypertonic solution. So water molecule will be moving out from the red blood cell into the environment. So by this, the red blood cell will shrink, change in size, we call crenation. While in the other way, for the plant cell, when I place my plant cell into a distilled water is hypotonic. By this, water molecule will move into the plant cell, makes the plant cell become turgid, but the plant cell won't burst because the plant cell have cell wall as a protective layer to prevent bursting of plant cell. Well, if I place the plant cell into a concentrated salt solution, which is a hypertonic, water molecule will move from inside of the plant cell to the outside. The diffusion of water from inside to outside makes the plant cell shrink. We call it as flaccid. However, because of presence of the cell wall, the plant cell won't change in size. But then, the plasma membrane and the cytoplasm will be pulled away from the cell wall and shrink. In this scenario, we say the plant cell is undergoing plasmolysis. This is another same scenario. In an isotonic solution meaning the solution, the concentration of solute in the external environment is equal to the concentration of solute inside the cell. By this, in isotonic solution, the cell remain their normal size. However, one thing you must know that in an isotonic solution, it doesn't mean no movement of water. Even though in isotonic solution, there is still water molecule moving in and out of the cell. However, the net movement is zero, which means in an isotonic solution, for example, you have 10 water molecule to moving into the cell. At the same time, you will also have 10 water molecule to moving out of the cell. So the net is zero. So the animal cell or the plant cell will retain their normal size and shape. However, in hypotonic solution, which means the solution outside is more diluted, has a lot of water compared to the cell concentration. By this, water molecule will move from the hypotonic solution in the surrounding, enter into the cell, make the cell to increase in size, and at the end, the red blood cell will burst. However, plant cell will undergoing turgid because they have cell wall to protect them. When you place the animal or plant cell in a hypertonic solution, which is a concentrated solution, water molecule will move from the inside of the cell to the surrounding of the cell. By this, animal cell will shrink and change size. We call crenation. While for the plant cell, they will be undergoing flaccid, which is the plasma membrane and the cytoplasm will be pulled away from the cell wall. This scenario is known as plasmolysis. The third type of passive transport is called facilitated diffusion. In facilitated diffusion, the term facilitated, which means they need help. In facilitated diffusion, is referred to the movement of particle from the region of higher concentration to the region of lower concentration. But this time, they require a carrier protein, a transport protein. However, there is no chemical energy required in this process because it is moving from high to low concentration, moving down the hill. Thus, no energy required. However, it needs a transport protein. Do you know the reason? This is because the particles are too large in size. For example, when I have amino acid or glucose, for example, the chamber in your left, I have a lot of red particle. The red particle is amino acid. So you can see the one in your left, you have a lot of red, amino acid, and the one in your right, you have very little red particle, amino acid. So amino acid will move from your left to your right. It is moving from the region high concentration amino acid to the region low concentration amino acid. It is known as along the concentration gradient. However, energy is not required. However, because amino acid they are too large in size, so they are unable to directly cross the gap between phospholipid. That's why they need a door, a chamber that is a carrier or transport protein. The role of transport protein is allow the large particle to directly moving through the plasma membrane. So, example of particles across the cell using facilitated diffusion, example like amino acid and glucose. And remember, they are always moved from high to low concentration. For the transport protein, we have two types of transport protein, and actually even we have more. Some are transport protein known as channel protein. Some are called carrier protein. The differences is very simple. Channel protein is always open. As long as the particle size can fit in the passage, then they are able to cross. However, carrier protein will open and close. For the particle to use carrier protein to enter into the cell, the particle must have molecular structure to fit into the binding site. Then only they are allowed to cross the cell. Now let's look at active transport. The term active means require energy. So active transport is defined as the transport of substances from a region lower concentration to a region higher concentration. In this scenario, energy is required. Why? Because in example, just imagine like you cycling up the hill. You are moving from the region low altitude to the region high altitude. You need to push a lot of energy. Then only you can moving up the hill. So this region is just representing the region of low concentration of solid particle. And this region representing the high concentration of solid particle. So for substances to move from region of lower concentration to the region of higher concentration, it is called against the concentration gradient. In this scenario, they need energy as they are moving from low to high. At the same time, they also need to have transport protein because they are unable to cross the phospholipid bilayer directly. So in summary, active transport have three significant terms. First, it's a movement of particle from low to high concentration. Second, it requires energy. Third, it requires transport protein. Example of the active transport that happening in our body is a sodium potassium pump. What is sodium potassium pump? All of our human body cell, we have sodium potassium pump. Before I talk about sodium potassium pump, let's look at the comparison between the passive and active transport. In the passive transport, it's always move more particle from region there where they are in a high concentration to the region where they are in low concentration. Just the differences between the simple diffusion and facilitated diffusions are: in simple diffusion, particle can move across the phospholipid bilayer directly because they are small in size. However, for facilitated diffusion, the particles are too big. They cannot pass through the phospholipid bilayer directly. They need a transport protein so that they are able to cross. However, both simple diffusion and facilitated diffusion, they are moving along concentration gradient. However, in the active transport, it's a movement of particle from the region where they are in low concentration to the region where they are in high concentration. Now it's moving against the concentration gradient. By this, ATP is required for them to move against concentration gradient. At the same time, they also need a transport protein. So example of simple diffusion is exchange of gases in alveolus. Example of facilitated diffusion is transport of glucose or amino acid across plasma membrane. While for osmosis, this term can only be used to describe water. Only water molecule can use the term osmosis. The rest particle, they cannot use the term osmosis. What example of active transport mostly are ions. An example is sodium potassium pump. Transport of sodium ion and potassium ion across the plasma membrane against concentration gradient. Now let's look at how sodium potassium pump acts to actively transport sodium and potassium in and out of the cell. This is an example of human cell. Inside the cytoplasm, you will find we have a lot of potassium ion, very little sodium ion. Outside the cell, there are a lot of sodium ion, very little potassium ion. So, for the sodium ion to move from cytoplasm out of the cell to the outside, it is moving from the region of low concentration to the region of high concentration. At the same time, for potassium ion to move from outside into the cell, it is moving from the region of low concentration to the region of high concentration. It is also against concentration gradient. By this process requires energy and require a transport protein. This transport protein is called sodium potassium pump. Sodium potassium pump, it can simultaneously transport sodium out of the cell, at the same time transport potassium ion into the cell. However, sodium potassium pump transport 3 sodium ion out of the cell, at the same time transporting 2 potassium ion into the cell. So you maybe notice that 3 sodium ion will be transported out of the cell, at the same time 2 potassium ion will be taken into the cell. The movement of both sodium and potassium ion, even though it's in different direction, however, if you look carefully, both of them are moving against concentration gradient from low to high concentration. Now, let's look at a little bit of detail. This is the cell, this is a cell, and this is a plasma membrane, and this is a sodium potassium pump. How the sodium potassium pump can transport 3 sodium out and 2 potassium in? First of all, 3 sodium ion will bind to the sodium potassium pump. Once 3 sodium ion bind to the sodium potassium pump, which facing inside the cytoplasm, this causes ATP molecule to break down to form ADP and phosphate. Energy is released. The release of energy allows the sodium potassium pump to change shape. Now, the sodium potassium pump is facing outside. By this, 3 sodium ion can release out of the cell. Now, at the same time, 2 potassium ion, which is at the outside of the cell, now will bind to its binding site in the sodium potassium pump. The binding of 2 potassium ion to the sodium potassium pump causes the phosphate, which is originally attached to the sodium potassium pump, to be detached. The removal of the phosphate group from the sodium potassium pump causes the sodium potassium pump return to their original position, which is facing inside. Now, 2 potassium ion can be released into the cytoplasm. And then the cycle will be repeat. 3 sodium ion again bind to the sodium potassium pump and then trigger the same process. Now you may be curious how the sodium potassium pump can know they should transfer how many sodium out of the cell and 2 potassium ion into the cell. The answer is very simple, just depend on the binding site. Because the sodium potassium pump have 3 binding sites for sodium, but only 2 binding sites for potassium. By this, in each transport, surely you will transport 3 sodium out of the cell and 2 potassium into the cell. We have already learned about passive transport and active transport. Now, let's look at endocytosis and exocytosis. Both endocytosis and exocytosis are purposely designed to transport large particles, in which the particle size are too big until they can't directly cross the transport protein. By this, they have to use endocytosis and exocytosis. So, endocytosis and exocytosis, they are active process, which means ATP energies are required during the process of transport. Now, let's look at exocytosis. Exo means exit. Cyto means cell. Exocytosis means exit from the cell, which means the cell are transporting particles out of the cell to the outside. In the exocytosis, first of all, the substance will be enclosed in a spherical structure called vesicle. The vesicle will fuse with the plasma membrane so that the particle can be directly removed out of the cell. This process is known as exocytosis. As you can see in this figure, the substances are enclosed in a vesicle. This vesicle moves towards the plasma membrane, and the vesicle will fuse with the plasma membrane to release the substances out of the cell. This is called exocytosis. While for endocytosis, endo means enter, Cyto means cell. So enters into the cell. We call them endocytosis. So simply means endocytosis is the cell taken up particles into the cytoplasm. Endocytosis can be divided into three types: phagocytosis, pinocytosis, and receptor-mediated endocytosis. All of them are under endocytosis, but the difference are in phagocytosis, it uptake large particle or solid particle, while in pinocytosis, it uptakes small particle, or we call them as liquid particle. So for example, in the phagocytosis, when the cell engulfs solid particles such as food particle, while in the pinocytosis, for example, the cell uptake liquid particle such as amino acid. You can see in this picture, in the phagocytosis, the cell is engulfing solid particle, their food particle. This is called phagocytosis. While pinocytosis is the cell uptake liquid particle such as amino acid. Now, how about receptor-mediated endocytosis? If you're comparing these three, phagocytosis and pinocytosis, they are non-selective, which means the cell will simply just uptake any solid or liquid particle. However, in receptor-mediated endocytosis, it uptake only specific particle. How can the cell differentiate the specific particle they need? This is based on the binding to the receptor protein on the surface of the cell. In receptor-mediated endocytosis, first of all, the specific substance need to bind to the receptor protein on the surface of membrane. If the binding is complementary, then only it will be uptake into the cell. The example is the process of uptake of cholesterol in the animal cell. This is the example. In the cell surface membrane, you have receptor protein. The substance will first bind to the receptor protein, and then the receptor protein bind to the substance, it will form into a vesicle. The vesicle will move into the cytoplasm and form into a substance to be transported into the cell. This is a comparison between three types of endocytosis. The phagocytosis, the cell uptake solid particle. The pinocytosis, the cell uptake liquid particle. And in receptor-mediated endocytosis, it uptake only specific particle. Those particles that can bind to the receptor protein, only they can be uptake into the cell. These are the comparison of different type of transport. In the next section, we're going to look at the concept of water potential. Now, what is water potential? Water potential is defined as the ability of water move out of the solution through osmosis. In this picture, as you can see, I have a U-shaped beaker or chamber. And this U-shaped structure, they are separated by a semi-permeable membrane. So, in this arm, it has lower concentration of solute. When you have lower concentration of solute, which means they have higher concentration of water. While in this arm, which is the one in your right, you have higher concentration of solute. Higher concentration of solute means have lower concentration of water. As such, water molecule will move from the region where they are in higher concentration of water, which is the one in the left, to the region where they are in lower concentration of water, which is the one in your right. So always remember, low concentration of solute, which means higher concentration of water. Higher concentration of solute, which means lower concentration of water. Now, we are having a cell. Inside the cell, it has water molecule. Water molecule tend to moving out from the cell. The concept of water molecule to move out of the cell is known as water potential of cell. It's defined as the water potential of cell is defined as the ability of water molecule to move out of cell by osmosis. Water potential always use a symbol psi. In the next picture, I have a beaker. Inside this beaker, it has solution. For example, sucrose solution 2 molar. Inside the solution, you have water molecule. The water molecule also tend to move out from the solution. The ability of water molecule to moving out from the solution is known as water potential for solution. So water potential for solution is the ability of water molecule to move out of the solution by osmosis. So in fact, water potential for solution and water potential for cell, they are similar, just differ in the origin. So for your level, what you need to know is only the definition for the water potential of cell. You do not need to know the definition for this one, the water potential for solution. This one do not need to know. Now, I have a beaker. Inside this beaker, it have added with solute particle. For example, this solute particle is sodium chloride, or it can be a sucrose, any solute particle. Now, outside, I have water molecule. The presence of solute particle in the solution will attract water molecule to go into the solution. The ability of this solute particle to attract water molecule to enter into the solution is known as solute potential. So solute potential, also known as osmotic potential, is defined as a measure of the effect of dissolved solute on the osmotic behavior of the solution. Simply, I can define them as the ability of the solute particle to attract water molecule to enter into the solution, which is defined as solute potential. So, high solute potential means high osmotic potential. Now, when I have a beaker separated by a semi-permeable membrane, and then the water molecule have ability to move through the solution, and if both solutions they are isotonic, means equal solute concentration, by this I will have net movement of the water molecule between the chamber will be 0, which means if I have 5 water molecules to move from left to the right, at the same time I will have 5 water molecules to move from the right chamber to the left. So the net movement will be 0 in isotonic solution. However, now if I have added solute particle in the solution, what happens is water molecule always they tend to bind to the solute particle. Once water molecule bind to the solute particle, the kinetic energy of water molecule decreases. By this, water potential of the water molecule also decreases. So always remember, when you have more solute particle in the solution, then more water molecule will bind to the solute particle. By this, the kinetic energy of the water molecule will decrease drastically. In conclusion, we can say for osmosis, it talk about the potential water molecule have to move. Now, distilled water is the condition whereby you do not have any solute particle at all in the solution. And we assign that the water potential of distilled water or the water potential of a pure water is 0 with the unit kilopascal. Now, this is a scenario whereby water molecule move freely the most. This is a scenario whereby water molecule has highest kinetic energy. Why? This is because in pure water or in distilled water, you do not have any solute particle at all. By this, in distilled water, this is a scenario whereby water molecule can move freely the most, highest kinetic energy or highest potential. Now, if I add a little bit of solute particle into the solution, what will happen? The kinetic energy of the water molecule decreases. Since water molecule have highest kinetic energy or highest potential is 0 kilopascal, once you add with solute particle, now the potential will decrease below 0, which is negative. That's the reason why water potential is always in negative value with the unit kilopascal. So high osmotic concentration means high solute potential, which means in high osmotic concentration you will have a lot of solute particle. When you have a lot of solute particle, then water molecule tend to bind to the solid particle. By this, water molecule cannot move freely, cannot move easily. The kinetic energy decreases, so the water potential decreases. So for your understanding, you can always equal the kinetic energy to water potential, which means low water potential means low kinetic energy of water. This is just simply for your understanding. So less negative of the water's potential, which means you have less solid particle, which means water molecule can move easily. The water molecule have more energy to move out. So remember, water molecule always move from less negative to more negative water potential. For example, now I have two cells, the one in your left and the one in your right. The cell in your left have water potential negative 0.5 kilopascal. The one in your right, they have negative 0.9 kilopascal. So can you tell me which cell has less solid particle? Left or right? The answer is left, because this cell has negative 0.5 kilopascal, which is less negative water potential. Less negative water potential means less solid particle. When you have less solid particle, which means less water molecule bind to the solute. So most of the water molecule in the cell can move freely. While in the more negative water potential, which means you have more solid particle. When you have more solid particle, most of the water molecule are bound to the solid particle, so they can't move freely. So in conclusion, the cell in your left, the water molecule can move freely since they have higher kinetic energy, since they have very little solid particle. So in conclusion, water molecule will always move from less negative cell to the cell with more negative. Now, this is a plant cell. Outside the plant cell, I have water molecule. Water, due to the presence of solid particle in the cell, this will attract water molecule to enter into the cell. When water molecule enter into the cell, the water molecule will make the plant cell become turgid. So this will try to push the plant cell to the cell wall. However, the plant have cell wall. The cell wall will resist the water molecule to push the plant cell from bursting. So what happens is the cell wall will give a revert pressure, known as pressure potential, to the cytoplasm to prevent the plant cell from burst. This revert pressure is known as pressure potential. Pressure potential defined as the pressure that derives from the cell wall to the cell to prevent the plant cell from bursting. Since the pressure potential is in opposite direction, so the pressure potential is always in positive value. So by this, I can summarize the three formula in an equation. The water potential of a plant cell is always equal to solute potential plus pressure potential. In summary, I have a cell. Inside this cell is a hypotonic cell. Hypotonic cell means a lot of water molecule, very little solute particle. So water molecule tend to move out from the hypotonic cell to a hypotonic cell. Now this is a hypertonic cell. Hypertonic cell have a lot of solid particle. So water molecule in a hypotonic cell, they tend to moving out. This is called water potential of a cell. The presence of solid particle in a hypertonic cell, will attract the water molecule to go in. This is called solute potential. Once water molecule go in, the water molecule will give a pressure to make the plant cell become turgid. So now the cell will give a reverse pressure to prevent from bursting. This reverse pressure is known as pressure potential. So this picture shows the relationship between water potential of cell, solute potential, and pressure potential.